everyone, welcome to my channel Pure Biology, and today I come with another video, that is the lysosome, and here we will learn about the structure and the function of the lysosome. So this is the part of the cell biology and it is the first topic related to cell biology. Today I will start. And before going to this topic, I want to say thank you to all of you who have already subscribed my channel, and also I request to the newcomers, please share this channel with your friends if this channel is helpful to you, okay, and obviously subscribe for the growth of this channel. So let's, and again I want to say that you just after subscription you try to press the bell icon for getting all notifications, and when I upload the video you will get the notification. So please do that, okay. So let's continue. This is the lysosome. What is the lysosome actually? You all know that lysosome is generally seen in the animal cell, okay. This is the organelle. First, what is cell? You know all that cell is the functional and the structural unit of the body, okay. Same functional cells that attach to each other to form the tissue, then same functional tissues they join to each other, then form the organ, and same type of functioning organs they attach to each other to form system. So in that way different systems that are seen in our body or in animals, okay. So lysosome is one kind of organelle that is seen generally in the animal cell, not in the plant cell. And this is called the suicidal bag. You know why this is called the suicidal bag? Because this contains many enzymes, okay, and its function is that it generally engulfs the external foreign particles and the internal foreign particles. Internal means within the cell and external means just outside the cell, okay. Its function is that just digest them, okay. So this is, you can say that this lysosome organelles, it is obviously related with the function of the macrophages and the neutrophils, etc., okay. So first we will see the structure. These are the single membrane bound organelle, okay. And another thing is, in the cell another thing I have to say that in the cell there are two parts. Generally one part that is the cytoplasm and another part that is the nucleus, okay. Nucleus is the you can say the master of the cell, or it controls the every functions of the cell, okay. So this is the master of the cell is the nucleus, and remaining part that is the cytoplasm, clear. And there are the organelles, many organelles that are seen within the cytoplasms, that are mitochondria, lysosomes, Golgi apparatus, another. And the cell is just protected by the cell membrane in animal cell, but obviously in plant cell there are another protective part that is called the cell wall, okay. This is the basic structure of the cell, okay. Now we will start about the lysosome. This is actually single membrane bound structure or organelle, and that is present in the animal cells, okay. These are heterogeneous. Heterogeneous means not are same all. That means the size and shape that always vary, okay, in case of lysosome, okay. All the lysosomes are not in same shape and size, okay. Next, these have acidic pH, okay. So within the lysosome the pH is acidic, and this is filled with hydrolytic enzymes. Now I have to say that why the internal portion of the lysosome, that is the pH of the lysosome, that is the acidic medium, because there are many enzymes. These are the hydrolytic enzymes. They act only in the acidic medium, okay, not or acidic environment, not in the alkaline environment. So the internal pH of the lysosome, that is acidic. What type of enzymes are there, that is proteases, nucleases, glycosidases, lipases, phospholipases, phosphatases and sulfatases. These kinds of enzymes are there within the lysosomes. These are responsible for the controlled intracellular digestion of macromolecules. Okay, these generally help in the digestion of macromolecules, if these are within the cell or just outside the cell. Okay, so I said that this line is saying that all are acid hydrolases because these require the acid environment for optimal activity, and lysosome provides this by maintaining pH about 5. Okay, so what I am saying that just before this, there are enzymes that act only the acidic environment. That's why internal environment of the lysosome that maintain the pH about 5. Okay, there are two types of lysosomes. One is primary, another is the secondary. What kind of primary and secondary? The secondary is that lysosome which containing the materials that are undergoing digestion or that already have been digested. Okay, so primary lysosome is without this material, but secondary lysosomes are those lysosomes that containing the materials that are either completely digested or just they are in the part of the digestion. Okay, lysosomal breakdown of materials are classified into two pathway, or you can say there are two type of breakdown is seen. One is the autophagy and another is the heterophagy. Okay, what is the autophagy? Autophagy means the degradation of the material. What kinds of materials or what kinds of components? That are cytoplasmic or within the cell, or the cell's component that are not necessary for the cell. These are degraded by this process. Clear? And heterophagy is that the process by which the substances are taken. Substances. These are taken into the cell from external environment. Clear? External environment and broken down by the lysosomes. Here the degradation occurring in two ways. One is the phagocytosis, another is the pinocytosis. Actually, phagocytosis is that what? The phagocytic cells are what? Which are macrophages or neutrophils. These engulf the large particles, and a single membrane phagosome is generated for this reaction, and this compartment may fuse directly with lysosome to produce phagolysosome. So what situation is happening here? First again understand, then you can learn. Okay, so what is that? Phagocytosis is what? Actually, some phagocytic cells which are macrophages or the neutrophils, these engulf the macromolecules or the large particles. Clear? And actually here one kind of cell produced, this is called the phagosome. This is obviously single membranous cell, or you can say the particle. This is called the phagosome, and within the phagosome these large particles remain here, and this phagosome fuse with the lysosome to form the phagolysosome. After fusing with the lysosome, these are completely degraded. Clear? So what is saying here, that macrophages or neutrophils, they engulf the large particles to destroy them. But after destruction of the large particles, what will happen to these macrophages or the neutrophils? These convert into phagosome, and these phagosome that containing with the degraded material fuse with the lysosome. form phagolysosome. Clear? And complete degradation is possible when the lysosome lysosome damage this totally. The second one is the pinocytosis. Here, generally the eukaryotic cells internalize fluid materials in small pinocytic vesicles. Okay. So, this process, or you can say pinocytic vesicles or the endocytic vesicles. So, here the particle that is formed with the degraded materials or fluid, these are known as the endocytosed substances, and the endocytosed substances are called which one? The fluid degrading particles or fluid degrading components. Okay, and cell that is formed that containing the degraded material, these are called the endosomes. Clear? These are called the endosomes, and then fuse with the lysosome. Lysosome. This is called the endolysosomes. I repeat again. First, the endocytic vesicles. These contain the degraded materials or the fluid particles. Clear? And these are called now endocytosed substances. And the endocytosed substances present within the vesicle, that is called the endosomes. And these endosomes when fuse with the lysosome, they form endolysosomes. And after that, the lysosome generally degraded the all try to degrade the all materials within that present within the endosomes. Clear? So these are the actual process occurring in the lysosome. And after that, we will try to the process autophagy. Here, this process is related to the heterophagy. Clear? And now we try to understand the autophagy process. Okay? Here, we just learned that the process very briefly, but heterophagy is explained properly here. And after this explanation, I will try to explain autophagy process in the lysosome. Clear? So continue with me. Okay. So in case of autophagy, the formation of phagophore is seen here. Okay. During autophagy, the formation of phagophore that begins, and these phagophores that form within the cytoplasm, the de novo, from a cup-shaped membrane that expands into a double membrane bound autophagosome. Okay. So what is happening here? These phagophores containing the substances form the autophagosome. Okay, autophagosome that having the double membranous structure. Just outside the autophagosome, there is double membranous structure, and or you can say that autophagosome is surrounding with a double membranous protection. You can say. Okay, so this autophagosome that fuse with the endosome, may fuse with an endosome, and this product is formed is autophagosome. So what will happen here? First, phagophores are formed during the autophagy. These phagophores that expands in shape and size, you can say, and form the autophagosome. Okay. And this autophagosome again fuse with the endosome, okay, to form autophagosome. Clear? This endosome fusion is called an amphisome. It is also called the amphisome. This amphisome fuses with the lysosome to form autolysosome. So here autolysosome is formed after fusion with the lysosome. So what is seen here? That phagophores first form, then phagophores expand in size to form autophagosome. These fuse with the endosome to form autophagosome or amphisome. These fuse with the lysosome to form autolysosome. Clear? And what is happening here after the degradation of the material within the lysosome? What is happening with the remnants, particles? Generally, some lysosome participate in exocytosis. So this enables the cells to eliminate these undigested contents. Clear? After complete digestion within the lysosome, there are remnants present within the cell, and these are just released from the cells with the help of obviously specific lysosome, and the process is called the exocytosis, by which this releases from the cell and just to the outside. And this is also possible by the lysosome, specific lysosome. Clear? And this is the rough structure of this is the cell, and this is the cytosol, this is plasma membrane. So here the three pathways just I have seen here, I just I have drawn here in three pathways how the lysosome just help to digestion, in digestion of the materials. The first part you can see that the process of the phagocytosis, here the large materials that is engulfed, and this totally attached with the lysosome before digestion, and these are the fluid molecules by the pinocytosis process that attach with the lysosome before digestion. And this process is the autophagosome process or autophagy process. Here also generally the autophagosomes, this is the complete part of the autophagosome, this attached with the lysosome before digestion. Okay, so here are the two steps, generally two pathways, not steps, two pathways. One is the heterophagy, this is the heterophagy, okay, and this is the autophagy. The two pathways just I have drawn here very you can say in rough sketch.